IB Cell Biology Part 1 will introduce some of the overarching themes of cell biology, including cell theory, the characteristics of living things that can be observed in single-celled organisms, and lastly, we'll look at stem cells. The essential idea is the evolution of multicellular organisms allowed cell specialization and cell replacement. Here is the outline of all the movies available for studying IB cell biology. Use this slide to reference the part of the unit that you need to review. This movie is focused right here. Biology can be studied at various levels, and we often deconstruct the subject in order to make it more manageable. Populations evolve in their natural environment as individuals live or die according to their reproductive success in that environment. The biology of individual organisms is an emergent property from the organs, the tissues, and the cells that make up multicellular organisms. And the functions of a single cell emerge from the molecules within. Biology can be studied at the ecological level, the population level, the individual organism level, the tissue level, cell level, or molecular level. Here's the first IB syllabus statement that guides this movie. Outline the cell theory and discuss the evidence for the cell theory. Then describe 3 exceptions to the cell theory, including striated muscle, giant algae, and aseptate fungal hyphae. Cell theory states that all plants and animals are composed of cells. As well, cell theory states that all cells have come from pre-existing cells. Schleiden studied plants, Schwann studied animals, and Virchow was responsible for linking cell theory to reproduction, a fundamental biological activity. Here's an outline of cell theory. All living things are composed of cells or cell products. Cells are the smallest units of structure and function in living things. All cells come from pre-existing cells. All the vital functions of an organism occur within a cell. We will look at the vital functions of an organism later in this movie. Pasteur provided evidence for cell theory by demonstrating that cells come from other cells. In other words, cells do not arise spontaneously. Pasteur boiled nutrient broth. He put the broth into flasks. One flask was open to the air where cells could enter, while the other flask was closed with bent glass tubing. The flask exposed to the air, exposed to cells, grew bacteria, while the flask without exposure to cells did not grow bacteria. Interestingly, there are organisms or cells that provide examples of discrepancies in cell theory, examples where cell theory needs to be modified. In biology, given the adaptable nature of organisms and their environment, there always seems to be exceptions to theoretical constructs. Striated muscle tissue. These cells are very long, and each cell has many nuclei. Are we looking at a single cell or many cells without membrane divisions between them? Again, exceptions or discrepancies in cell theory might include giant algae, whose cells are huge. While we might expect such large structures to be multicellular, they are not, as they house just a single nucleus. Fungi often have long thread-like structures known as hyphae that have many nuclei in very long cells. Again, as an exception to cell theory, we might expect many smaller cells in hyphae, but instead we see long, undivided sections that contain many nuclei. The next IB syllabus statement for this introduction to cell biology is: state that unicellular organisms carry out all the functions of life: metabolism, response, homeostasis, growth, reproduction, and nutrition. Here are the characteristics of all living organisms. To be considered alive, all of these criteria must be met. Organisms grow and develop, they contain a genetic code, they reproduce, respond to the environment, exchange energy and matter with the environment, they're made up of cells, and they display homeostasis. I remember this list as GREECH, G-G-R-R-E-C-H, grow, genetic code, reproduce, respond to the environment, exchange energy and matter, cells, and homeostasis. Let me provide some examples of one-celled organisms that carry out all the functions of life, and I'll start with nutrition. The paramecium, the image on the left, is a heterotroph, also known as a consumer. The paramecium feeds on living organisms by ingestion. The buccal cavity is here, and food enters the buccal cavity, and at the end of the cavity, food is taken in by endocytosis. These are food vacuoles, and the food is digested by enzymes contained within the cell. The Chlamydomonas, the image on the right, is an autotroph. Autotrophs obtain inorganic nutrients from the environment and synthesize organic molecules, for example, in the process of photosynthesis. Growth. The paramecium gets nutrients from ingested food, and these nutrients provide both carbon and energy for growth in size. The Chlamydomonas absorbs inorganic molecules directly from the environment to provide for growth. Photosynthesis, for example, is a process that synthesizes energy and carbon-rich organic molecules from inorganic molecules. This allows the Chlamydomonas to grow. Excretion. In both organisms, some wastes diffuse directly out of the cell across the membrane. In paramecium, carbon dioxide diffuses out, while other wastes are excreted through exocytosis. This is a contractile vacuole that expels water to maintain homeostasis. In Chlamydomonas, oxygen, a waste product of photosynthesis, diffuses directly out of the cell. Carbon dioxide, a waste product of cell respiration, also diffuses directly out of the cell. And then there's response to stimulus. The paramecium uses cilia to move the organism toward or away to or from external stimuli. The Chlamydomonas uses an eye spot and flagella to move the organism toward light. Metabolism is the sum total of all chemical reactions in the body. All reactions in living organisms are catalyzed by enzymes. The paramecium has cytoplasmic enzymes that catalyze reactions such as the early stages of cell respiration. The paramecium also has a mitochondrion, which has enzymes that catalyze reactions as a part of cell respiration. In Chlamydomonas, enzymes in the chloroplast catalyze photosynthesis, while enzymes in the mitochondria catalyze cell respiration. Homeostasis is the process by which a constant internal environment is maintained despite fluctuating external conditions. In other words, body functions are maintained within narrow limits. For example, as a mammal, your body has mechanisms to maintain body temperature within narrow limits. The paramecium and the Chlamydomonas also have homeostatic mechanisms. For example, these organisms use contractile vacuoles, seen here, to remove water, thus maintaining osmotic balance, water balance. The level of water within the cell is maintained within narrow limits by the use of the contractile vacuole. And then there's reproduction. The paramecium and the Chlamydomonas use asexual cell division, known as mitosis, and sexual cell division to produce gametes, sperm and egg, that fuse through fertilization. While I've compared the paramecium as a heterotroph with Chlamydomonas as an autotroph, there are organisms such as Euglena that are both. Euglena can ingest other organisms like the paramecium, and it has chloroplasts for photosynthesis like Chlamydomonas. Single-celled organisms carry out many metabolic processes, all within the confines of a single cell. For example, single-celled bacteria can conduct photosynthesis, chemosynthesis, respiration, anaerobic respiration, and nitrogen fixation. Here's the next IB syllabus statement. Explain that cells in multicellular organisms differentiate to carry out specialized functions by expressing some of their genes, but not others. Cells within us, while each carrying the same genetic composition, are specialized for different functions, as you can see here in this image. In each cell separately, some genes are expressed, while other genes are not. The expression of genes differs among all the differentiated cells in our body. Again, every cell in our body has the same complement of DNA, but depending on the cell type, some genes are expressed, turned on, while others are not expressed, turned off. The cell can regulate gene expression through various mechanisms, which we will study later in this course. Earlier in the movie, I mentioned how the activities of an entire organism emerge from the organs, tissues, and cells of which it is made. The idea here is called emergent properties. Take note, emergent properties. The idea that the whole is greater than the sum of its parts. The handle of the hammer is not useful by itself. The head of the hammer is not useful by itself, but together, the handle and the head together, a useful structure emerges. You can see the idea of emergent properties in this diagram, an idea that I've already layered into this movie, that something living requires many properties, many characteristic properties all together. Taking in nutrition or excreting waste or responding to stimulus are not enough if the organism cannot reproduce or the organism cannot maintain internal homeostasis. Life emerges from the component parts and all of the component processes. The heart is a pump, nothing more than a pump, but the heart is composed of cells, and the pump emerges from the collection of cells, each one of which alone could not pump the blood of an animal. Thinking and reflection emerges from a collection of nerve cells. A tree, as an individual organism, successful in its environment, emerges from a collection of cells, some of which are not photosynthetic and some of which are. The non-photosynthetic cells absorb water and nutrients from the soil, while the photosynthetic cells absorb carbon dioxide from the air. Anabaena is a filamentous bacterium composed of some cells that are photosynthetic and some, about 1 out of 10, that can fix nitrogen from the air. The whole organism emerges from the collection of both cell types. The whole is better than the parts alone. The termite mound emerges from the colony of individual termites. The focus of emergent properties, as I've presented it here, has been that multicellularity is more than just a collection of many cells. The activities of a successful multicellular organism emerge from a collection of individual cells, any one of which alone could not display. Throughout this course, we'll be looking at many examples of emergent properties. And the last IB syllabus statements of this movie are: outline that stem cells retain the capacity to divide and have the ability to differentiate along different pathways. Outline the therapeutic use of stem cells to treat Stargardt's disease, and one other named example. Some cells in the adult body remain open or pluripotent as stem cells. Bone marrow, skin, fat tissue. There's great potential for tissue repair, treating degenerative disease such as Parkinson's or other diseases. Stay tuned. Stem cells are undifferentiated cells, but they're important because they have the capacity to divide and differentiate along different pathways. We will see in the slides to come the use of stem cells to treat disease. The handful of cells that comprise an early embryo are stem cells. These cells could be harvested, as you can see here, and cultured to differentiate into a tissue that might repair tissues that are damaged. Stem cells, as undifferentiated cells, are open to differentiation. They can be cultured to differentiate such that they can be used for therapy, leukemia, or Stargardt's disease, which is a disease of the eye. If diseased tissues can be replaced by healthy tissues derived from stem cells, much suffering can be alleviated. Stargardt's disease results from a genetic mutation. There's a loss of vision beginning about the ages of 6 to 12, and in 2010, stem cells were implanted in an adult woman in a 4-month trial, and vision was improved. Here's an IB syllabus statement that follows up on stem cell use. Discuss the ethics of therapeutic use of stem cells taken from specially created embryos, from the umbilical cord blood of a newborn, and from an adult's own tissues. While embryos are sources of stem cells, harvesting stem cells from embryos is hugely controversial. Should 8-celled embryos be considered living to the extent that they have rights under the law, and to that extent should they be used to provide stem cells for people who suffer from disease? But if we could use embryonic stem cells, a lot of suffering could be alleviated. In vitro fertilization, the fusion of sperm and egg in a petri dish outside the body, results in an embryo that could be used to produce stem cells that results in new specialized tissues. Is this ethically acceptable? Here's an outline of the arguments around using embryos as a source of stem cells. The argument against would be that an embryo is life no matter what. But in favor of using embryos would be that early stage embryos have yet to develop, and early stage embryos do not feel pain, and deliberate embryo production is not denying what would have been a life, and they can help to reduce suffering. There are other sources of stem cells, from the placenta or from the umbilical cord, specifically the blood from the umbilical cord. The use of placental stem cells would remove the ethical concerns of using embryos. Scientists are working on harvesting stem cells from the body of the person requiring new tissue or requiring transplanted tissue. Skin cells, known as fibroblasts, can be cultured into stem cells, which then can be differentiated into different cell types and used to replace diseased tissue. In fact, new techniques are being developed where the nucleus of a differentiated cell of a patient is placed into the egg that's had its nucleus removed. The embryo formed from this technique could be used to develop stem cells that result in new tissues that are transplanted back into the patient. Consider how ethical concerns shape governmental restrictions on stem cell use, and how governments are influenced by cultural and religious traditions. In terms of the arguments against the use of embryonic stem cells, the primary argument is the source of stem cells. Embryos provide stem cells, and using embryos would require killing a life. Every embryo should be given a chance of developing. Embryonic stem cells could develop into tumor cells. And stem cell source is limited, so it's expensive. It wouldn't be available to everyone. The arguments in favor of stem cell use would be that stem cells can be used to treat disease called therapeutic cloning, and this reduces suffering. Stem cells can be used to build new tissues as a source for transplants, and stem cells could have genes inserted, and then these cells are then injected into the tissue that require a normal gene rather than a faulty gene, and this is called gene therapy. And that brings us to the end of Cell Biology Part 1. Cell Biology Part 2 continues with an introductory look at ideas in cell biology, including the surface area to volume ratio concept.